This video is going to go over the case study, the malaria case study that we've spent the past few days on. It's going to include a statistical analysis and also a focus on the allele frequency of a population over time. This graph, or this table, this map, is what sort of sparked this case study. When we look at the distribution of the sickle cell allele and we sort of superimpose that on top of malaria incidences, we can see that in regions that are highly susceptible to malaria infection, either in certain seasons or throughout the entire year, in these regions we see an abnormally high frequency of the sickle cell allele. This holoendemic color essentially means that the malaria parasite is present in every month out of the year. You have to remember that the malaria parasite relies on mosquitoes for transmission, and this holoendemic category means that the mosquito, the Anopheles mosquito, is present every month of the year in these regions and therefore is able to transmit the malaria parasite to an enormous number of individuals who live in these specific areas. And so the pattern that sparked this case study was the pattern of high rates of malaria and very persistent malaria persistent regions also are regions that have very high frequencies of the sickle cell allele relative to frequencies in other parts of the world. So very high frequencies here relative to the sickle cell allele frequency in North America. So let's look at why that might be. And first we're going to start and look at what exactly an allele is. So an allele is simply a variant of a gene. Well, let's make sure we understand what a gene is. A gene, right now, is a segment of DNA that is providing instructions for making a protein. And remember that the letters of DNA are A, T, G, and C. So these sequence of letters tell a cell how to make a specific type of protein. So for example, this sequence right here is telling a cell to use the amino acid leucine followed by the amino acid histidine. Now, you don't have to know that right now. My point is that the sequence of the DNA letters is ultimately going to determine what the primary structure of a protein looks like. And so if you change the sequence of DNA letters may result in a change in the primary structure of the protein. And in this case, it would. Changing these letters from AAC would result in not leucine, but phenylalanine, a different amino acid being used when this polypeptide is being created. Now we know from chapter 5 that the primary structure of a polypeptide ultimately is going to determine how that polypeptide folds. And we know from our hemoglobin activity that changing the primary structure of the beta subunit in hemoglobin Specifically, what we're changing is glutamic acid is being replaced by valine. When the primary structure is changed, what we end up with is a hemoglobin molecule, which remember is composed of 4 subunits. 2 of those subunits are beta subunits, the other 2 are the alpha subunits. What we end up with are the beta subunits have a hydrophobic R group poking out, which results in a cable formation of hemoglobin molecules. So the point that I'm trying to make here is that changing the letter can result in a change in the protein's shape and therefore a change in the protein's function. So if a gene is a segment of DNA that provides instructions for making a protein, and the gene that I'm talking about right here in this example is the gene for beta subunit protein. Remember, the beta subunit is an actual polypeptide. So it's composed of amino acids. The beta subunit gene is around 1600 letters of DNA long. So 1600 letters of A's, T's, G's, and C's in a very specific sequence. So let's represent that stretch of DNA by doing something like this. Let's say this is my hemoglobin gene, and this is the first letter, and it goes all the way 1600 letters later and ends right here with the last letter. What we were focusing on in class is a very specific letter. Let's just say this letter right here happens to be A. Now, if that letter is A, what you end up with is the production of a normal beta subunit, and we call that HbA. If you happen to have a gene, the hemoglobin gene, if you happen to have the hemoglobin gene that has a T, has the letter T instead of the letter A, what you end up with is a slightly different beta subunit, and that beta subunit, as you probably already know, is the sickle cell beta subunit. This single letter change right here, changing it from A to T, is the difference between glutamic acid up here and valine down here. So these are both genes for the beta subunit, but they are different versions of the same gene. So we call variations, variants, or different versions of the same gene, we refer to those as alleles. So an individual who has HbA, because remember you have one gene that you inherit from mom and one gene that you inherit from dad. This individual has both of their genes are this sequence or segment of DNA. An individual who is HbA, HbS has one gene for hemoglobin that looks like this, and their other gene for hemoglobin looks like this. So they have inherited one copy that produces normal beta subunit hemoglobin and one copy that produces sickle cell hemoglobin. And then finally, the other possibility is that the individual can be HbS, and if that's the case, then both of their copies of the hemoglobin gene are the sickle cell hemoglobin variation. So now that you're comfortable with what a gene is and what an allele is, let's look at a scenario where you inherit these genes from your parents. So your mother is a carrier for the sickle cell trait, so she is HbA, HbS, and your father is also a carrier for the sickle cell trait. That just makes things more interesting. And when they reproduce and create you, they are passing down one of their alleles to their offspring to make you. And which allele they pass down, either the HbA or the HbS, is completely random. It is a flip of a coin. So let's say this is heads and this is tails. And so we flip a coin and we see that the mother contributes or donates the HbA allele to the offspring. So this baby has HbA from mom. Actually, I'll draw it in red so we know that it came from mom. So HbA from mom. And now we do a flip of the coin, heads or tails. It is going to be tails. So this child is inheriting the sickle cell allele from the father. So this is an individual who is a carrier. Now, most of you, if you're remembering your middle school biology and you remember talking about alleles, most of the alleles were either capital or lowercase letter, and the capital letter was the dominant allele and the lowercase letter was the recessive allele, and you were probably taught that the recessive allele is recessive because it's not being expressed. So if you operate under that assumption, you might think that, okay, well, maybe this is the dominant allele and this is the recessive allele, so this person, this green individual, would have or would appear perfectly normal, much like an individual who looked like this would appear perfectly normal in terms of their hemoglobin. But that's actually not the case. That's not what's happening here. Actually, both of these alleles are being expressed simultaneously. They're both being read. So what that means is that if you look in the red blood cell of this green individual, there's our dumbbell-shaped red blood cell, you actually find some hemoglobin proteins that are normal. They have the glutamic acid, and you also find some hemoglobin proteins that are the sickle cell hemoglobin proteins. They have valine in their 6th amino acid position instead of glutamic acid. So this is an example of what's called co-dominance, which means that both of the alleles are equally being expressed. So they're both dominant instead of one being dominant and one being recessive. So this is a carrier. Carriers do not typically do not display symptoms. I say typically because they can experience symptoms of sickle cell anemia under extreme conditions, like for example dehydration and high elevation. So dehydration and elevation can play a role in triggering the sickling of a red blood cell, but without these extreme conditions, there is not as nearly as much red blood cell sickling occurring compared to an individual. Let's say they have another child over here. They have another child, and this child happens to inherit HbS from mom and HbS from dad. This individual would have red blood cells that look like this, and all of the hemoglobin proteins would be the sickle cell hemoglobin, and the result would be much more extensive sickling of the red blood cells compared to this individual over here. And this individual would have no sickling of the red blood cells because all of their hemoglobin proteins are normal.